A brain-computer interface, sometimes called a mind-machine interface, direct neural interface, or brain-machine interface, is a direct communication pathway between an enhanced or wired brain and an external device. BCIs are often directed at researching, mapping, assisting, augmenting, or repairing human cognitive or sensory motor functions. Research on BCIs began in the 1970s at the University of California, Los Angeles under a grant from the National Science Foundation, followed by a contract from DARPA. The papers published after this research also marked the first appearance of the expression brain-computer interface in scientific literature. The field of BCI research and development has since focused primarily on neuroprosthetics applications that aim at restoring damaged hearing, sight and movement. Thanks to the remarkable cortical plasticity of the brain, signals from implanted prostheses can, after adaptation, be handled by the brain like natural sensor or effector channels. Following years of animal experimentation, the first neuroprosthetic devices implanted in humans appeared in the mid-1990s. History. The history of brain-computer interfaces starts with Hans Berger's discovery of the electrical activity of the human brain and the development of electroencephalography. In 1924, Berger was the first to record human brain activity by means of EEG. Berger was able to identify oscillatory activity, such as Berger's wave or the alpha wave, by analyzing EEG traces. Berger's first recording device was very rudimentary. He inserted silver wires under the scalps of his patients. These were later replaced by silver foils attached to the patient's head by rubber bandages. Berger connected these sensors to a Littmann capillary electrometer, with disappointing results. However, more sophisticated measuring devices, such as the Siemens double-coil recording galvanometer, which displayed electric voltages as small as 1/10,000th of a volt, led to success. Berger analyzed the interrelation of alternations in his EEG wave diagrams with brain diseases. EEGs permitted completely new possibilities for the research of human brain activities. Professor Jacques Vidal coined the term BCI, and produced the first peer-reviewed publications on this topic. Vidal is widely recognized as the inventor of BCIs in the BCI community, as reflected in numerous peer-reviewed articles reviewing and discussing the field. Vidal's first BCI relied on visual evoked potentials to allow users to control cursor direction, and visual evoked potentials are still widely used in BCIs. After his early contributions, Vidal was not active in BCI research, nor BCI events such as conferences, for many years. In 2011, however, he gave a lecture in Graz, Austria, supported by the Future BNCI project, presenting the first BCI, which earned a standing ovation. Vidal was joined by his wife, Laras Vidal, who previously worked with him at UCLA on his first BCI project. Professor Vidal will also present a lecture on his early BCI work at the 6th Annual BCI meeting, scheduled for May-June 2016 at Asilomar, California. BCI versus neuroprosthetics. Neuroprosthetics is an area of neuroscience concerned with neural prostheses, that is, using artificial devices to replace the function of impaired nervous systems and brain-related problems, or of sensory organs. The most widely used neuroprosthetic device is the cochlear implant, which, as of December 2010, had been implanted in approximately 220,000 people worldwide. There are also several neuroprosthetic devices that aim to restore vision, including retinal implants. The difference between BCIs and neuroprosthetics is mostly in how the terms are used. Neuroprosthetics typically connect the nervous system to a device, whereas BCIs usually connect the brain with a computer system. Practical neuroprosthetics can be linked to any part of the nervous system, for example, peripheral nerves, while the term BCI usually designates a narrower class of systems which interface with the central nervous system. The terms are sometimes, however, used interchangeably. Neuroprosthetics and BCIs seek to achieve the same aims, such as restoring sight, hearing, movement, ability to communicate, and even cognitive function. Both use similar experimental methods and surgical techniques. Animal BCI research. Several laboratories have managed to record signals from monkey and rat cerebral cortices to operate BCIs to produce movement. Monkeys have navigated computer cursors on screen and commanded robotic arms to perform simple tasks simply by thinking about the task and seeing the visual feedback, but without any motor output. In May 2008, photographs that showed a monkey at the University of Pittsburgh Medical Center operating a robotic arm by thinking were published in a number of well-known science journals and magazines. Other research on cats has decoded their neural visual signals. Early work in 1969, the operant conditioning studies of Fetz and colleagues at the Regional Primate Research Center and Department of Physiology and Biophysics, University of Washington School of Medicine in Seattle, showed for the first time that monkeys could learn to control the deflection of a biofeedback meter arm with neural activity. Similar work in the 1970s established that monkeys could quickly learn to voluntarily control the firing rates of individual and multiple neurons in the primary motor cortex if they were rewarded for generating appropriate patterns of neural activity. Studies that developed algorithms to reconstruct movements from motor cortex neurons, which control movement, date back to the 1970s. In the 1980s, Apostolis Georgopoulos at Johns Hopkins University found a mathematical relationship between the electrical responses of single motor cortex neurons in rhesus macaque monkeys and the direction in which they moved their arms. He also found that dispersed groups of neurons, in different areas of the monkeys' brains, collectively controlled motor commands, but was able to record the firings of neurons in only one area at a time, because of the technical limitations imposed by his equipment. There has been rapid development in BCIs since the mid-1990s. Several groups have been able to capture complex brain motor cortex signals by recording from neural ensembles and using these to control external devices. Prominent research successors Kennedy and Yang Dan. Philip Kennedy and colleagues built the first intracortical brain-computer interface by implanting neurotrophic cone electrodes into monkeys. In 1999, researchers led by Yang Dan at the University of California, Berkeley decoded neuronal firings to reproduce images seen by cats. The team used an array of electrodes embedded in the thalamus of sharp-eyed cats. Researchers targeted 177 brain cells in the thalamus lateral geniculate nucleus area, which decodes signals from the retina. The cats were shown 8 short movies, and the neuron firings were recorded. Using mathematical filters, the researchers decoded the signals to generate movies of what the cats saw and were able to reconstruct recognizable scenes and moving objects. Similar results in humans have since been achieved by researchers in Japan. Miguel Nicolelis, a professor at Duke University, in Durham, North Carolina, has been a prominent proponent of using multiple electrodes spread over a greater area of the brain to obtain neuronal signals to drive a BCI. After conducting initial studies in rats during the 1990s, Nicolelis and his colleagues developed BCIs that decoded brain activity in owl monkeys and used the devices to reproduce monkey movements in robotic arms. Monkeys have advanced reaching and grasping abilities and good hand manipulation skills, making them ideal test subjects for this kind of work. By 2000, the group succeeded in building a BCI that reproduced owl-monkey movements while the monkey operated a joystick or reached for food. The BCI operated in real time and could also control a separate robot remotely over Internet Protocol. But the monkeys could not see the arm moving and did not receive any feedback, a so-called open-loop BCI. Later experiments by Nicolelis using rhesus monkeys succeeded in closing the feedback loop and reproduced monkey reaching and grasping movements in a robot arm. With their deeply cleft and furrowed brains, rhesus monkeys are considered to be better models for human neurophysiology than owl monkeys. The monkeys were trained to reach and grasp objects on a computer screen by manipulating a joystick, while corresponding movements by a robot arm were hidden. The monkeys were later shown the robot directly and learned to control it by viewing its movements. The BCI used velocity predictions to control reaching movements and simultaneously predicted hand gripping force. In 2011, O'Doherty and colleagues showed a BCI with sensory feedback with rhesus monkeys. The monkey was brain controlling the position of an avatar arm while receiving sensory feedback through direct intracortical stimulation in the arm representation area of the sensory cortex. Donahue, Schwartz and Anderson. Other laboratories which have developed BCIs and algorithms that decode neuron signals include those run by John Donahue at Brown University, Andrew Schwartz at the University of Pittsburgh, and Richard Anderson at Caltech. These researchers have been able to produce working BCIs, even using recorded signals from far fewer neurons than did Nicolelis. Donahue's group reported training rhesus monkeys to use a BCI to track visual targets on a computer screen with or without assistance of a joystick. Schwartz's group created a BCI for three-dimensional tracking in virtual reality and also reproduced BCI control in a robotic arm. The same group also created headlines when they demonstrated that a monkey could feed itself pieces of fruit and marshmallows using a robotic arm, controlled by the animal's own brain signals. Anderson's group used recordings of pre-movement activity from the posterior parietal cortex in their BCI, including signals created when experimental animals anticipated receiving a reward. Other research, in addition to predicting kinematic and kinetic parameters of limb movements, BCIs that predict electromyographic or electrical activity of the muscles of primates are being developed. Such BCIs could be used to restore mobility in paralyzed limbs by electrically stimulating muscles. Miguel Nicolelis and colleagues demonstrated that the activity of large neural ensembles can predict arm position. This work made possible creation of BCIs that read arm movement intentions and translate them into movements of artificial actuators. Carmina and colleagues programmed the neural coding in a BCI that allowed a monkey to control reaching and grasping movements by a robotic arm. Lebedev and colleagues argued that brain networks reorganized to create a new representation of the robotic appendage in addition to the representation of the animal's own limbs. It is conceivable, or even likely, however, that such a sensor will be developed within the next 20 years. The use of such a sensor should greatly expand the range of communication functions that can be provided using a BCI. Development and implementation of a BCI system is complex and time-consuming. In response to this problem, Gerwin Schalk has been developing a general-purpose system for BCI research, called Bci 2000. Bci 2000 has been in development since 2000 in a project led by the Brain-Computer Interface R&D program at the Wadsworth Center of the New York State Department of Health in Albany, New York, USA. A new, wireless approach uses light-gated ion channels such as channel rhodopsin to control the activity of genetically defined subsets of neurons in vivo. In the context of a simple learning task, illumination of transfected cells in the somatosensory cortex influenced the decision-making process of freely moving mice. The use of BMIs has also led to a deeper understanding of neural networks and the central nervous system. Research has shown that despite the inclination of neuroscientists to believe that neurons have the most effect when working together, single neurons can be conditioned through the use of BMIs to fire a pattern that allows primates to control motor outputs. The use of BMIs has led to development of the single neuron insufficiency principle, which states that even with a well-tuned firing rate, single neurons can only carry a narrow amount of information, and therefore the highest level of accuracy is achieved by recording firings of the collective ensemble. Other principles discovered with the use of BMIs include the neuronal multitasking principle, the neuronal mass principle, the neural degeneracy principle, and the plasticity principle. The Bci Award, the annual Bci Research Award, endowed with $3,000 is awarded in recognition of outstanding and innovative research in the field of brain-computer interfaces. Each year, a renowned research laboratory is asked to judge the submitted projects and to award the prize. The jury consists of world-leading BCI experts recruited by the awarding laboratory. Following list consists the winners of the Bci Award 2010. Kuntae Guan, Kai Keng Ang, Karen Sui Gio Kachua and Beng Tiang. Motor imagery-based brain-computer interface robotic rehabilitation for stroke. 2011. Moritz Grosswentrup and Bernhard Skolkoff. What are the neurophysiological causes of performance variations in brain-computer interfacing? 2012. Sergio R. Sokardar and Niels Berbaumer. Improving efficacy of Bci lesional brain-computer interface training in neurorehabilitation of chronic stroke. 2013. MC. Dardalata. B. E. Odokatia. N. Sabiza. B. A learning-based approach to artificial sensory feedback. Intracortical microstimulation replaces and augments vision. 2014. Katsuhiko Hamada, Hiromu Mori, Hiroyuki Shinoda, Thomas M. Rutkowski.